This video describes both manual and automated purification of circulating cell-free DNA from plasma samples. The MagMax cell-free DNA isolation kit may also be used to isolate cell-free DNA from serum and urine. Throughout the process, magnets are utilized to isolate the DNA, making this protocol simple and scalable. First, separate the cells from the plasma sample. Centrifuge the blood collection tube at 2000 G for 10 minutes and transfer the plasma to a fresh tube. Centrifuge again at 16,000 G for 10 minutes and transfer the cleared plasma to the fresh tube. Manual isolation can be completed in less than 45 minutes for up to 8 samples. To get started, add 2.5 mL of the lysis binding solution and 30 microliters of My1 silane beads to a fresh conical tube and mix by vortexing. Add 2 mL of plasma sample, mix by vortexing, and incubate for 10 minutes while shaking or rotating. Pellet the magnetic beads using a Dynamag 50 magnetic stand. Discard the supernatant as biological waste. Add 1 ml of wash solution, vortex for 30 seconds. And transfer the bead slurry to a microcentrifuge tube in a Dynamag 2 magnetic stand. Let stand 1 to 2 minutes or until solution clears and the beads are pelleted against the side of the tube. Carefully remove the supernatant, ensuring you do not disturb the bead pellet. Now add 1 ml of wash solution. Vortex for 30 seconds and briefly centrifuge to remove liquid from the cap. Again, place the tube on the Dynamag 2 magnet for 1 to 2 minutes and remove supernatant. Add 1 ml of 80% ethanol, vortex, and briefly centrifuge. Repeat the 80% ethanol wash process. Place the tube on the Dynamag 2 magnet for 1 to 2 minutes, leaving the tube on the magnet. Remove the supernatant, tap the magnet stand down 5 times, and remove any residual liquid. Let air dry for 3 to 5 minutes. Now add 50 microliters of DNA elution solution. Close cap and tap the tube. Vortex for 5 minutes and briefly centrifuge. Place tube in the Dynamag 2 magnet for 2 minutes or until the solution clears and the beads are pelleted. Your purified cell-free DNA is ready to use immediately, or the solution can be transferred to a non-stick microcentrifuge tube for storage. Place the tube at -20 degrees Celsius for long-term storage. Circulating cell-free DNA isolation may be automated using the Kingfisher Flex or the Kingfisher Duo magnetic particle processor. Use the table provided in the product manual to prepare the processing plates. Gently shake plate 1 to mix the reagents. Add your plasma samples to rows A and B according to the table. Ensure that the instrument is set up for processing with the deep well magnetic head and select the program on the instrument according to the table. Start the run. Load the prepared processing plates when prompted. At the end of the run, remove the plates and transfer the eluted cell-free DNA to an elution plate. Your purified cell-free DNA is ready to use immediately. For long-term storage, seal the plate or transfer the solution to a non-stick microcentrifuge tube and place at -20 degrees Celsius.